Wonderful to have you all here. I'm Jose Flores, as was mentioned. We have, for the next 40 minutes or so, this terrific conversation to have with an esteemed panel of experts, Drs. Rox Anderson, John Lepore, Jean-Francois Formella, and Stephen Greenberg. They span both clinical innovation from the academic setting, but also lots of experience in investment and in industry. So I think we'll have a very lively conversation. And of course, Nicole Davis will also tell us about the process that brought in all the brain power of Harvard Medical School, dozens of experts trying to come up with this list. Now we have 40 minutes to deal with 10 big, large unmet clinical needs with 4 additional runner-ups that you will see listed. And so 40 minutes is really not enough to be able to have 4 people comment on each one. So instead, we have a little bit of a different strategy that I think will make it for a more lively conversation.
Thank you, Jose, and welcome. Big thank you to Chris Coburn, to Nicole, and to all the folks who have planned not only the WMIF, but also this session. Very honored and excited to be part of it. Again, my name's Bruce Levy. In thinking about framing this session, I'm reminded of the masterpiece by René Magritte called Clairvoyance, where the painter is sitting in a chair, and next to him on the table is an egg. But he's painting a bird in flight, thinking about the transformation of the present to the future. Clairvoyance. Today, we're so fortunate to have these experts with us to think about this and to have a discussion on this topic. Nicole Davis led a team that did remarkable work in identifying these themes that we'll be discussing today to facilitate our session. So I definitely want to pass the mic, if you will, over to Nicole to tell us a little bit more about the effort that led to today's theme.
Thanks a lot, Bruce. Something we heard yesterday in Secretary Kate Walsh's fireside chat really resonated with me and I think really reflects the intent of what we were trying to accomplish here. She said, you can't fix what you don't see. And so we really set out to ask practitioners, what do you see? What needs to be fixed? And so we engaged a broad set of clinicians and scientists across MGB and asked them, what are the major unmet needs that they see every day? And, these are deeply expert and passionate people. They're on the front lines. And so it was a real pleasure to speak with them. And I just want to pause and thank everyone who participated in these discussions across the months of interviews, as well as the folks who helped us review data and our analysis process, as well as select and rank the final list that you see behind me. So as mentioned, I spoke with over 100 MGB Harvard faculty, actually 104 to be exact. And depending on your perspective, that may be a big number or a not so big number. But what was interesting is that it was sufficiently large to see some patterns. And by patterns, I mean people speaking about the same or highly similar unmet needs. And although, speaking about the individual unmet needs that came through this process would take a huge conference and way more time than we have. There was a lot of richness of information that came through the process. And so I just want to pause and take a moment and talk about the ones that were most often cited to me through these discussions. So the big 3 were early detection of disease, particularly cancers, but also other diseases such as neurodegenerative disease. Brain health was a very big theme, and the need to create a new framework for keeping the brain healthy and understanding really what that means, as well as diagnosing and treating diseases, especially Alzheimer's disease, but really other conditions that span the brain as well. And then topping out the big 3, new approaches to antibiotic resistance. This is not a new problem. We've known about it, but it's not going away. And the people that I spoke with were very concerned because it seems to be getting worse. And then the final sort of big 3, the next set of big 3, geriatric medicine. We heard in the talk yesterday from Hayam Israel about the aging of the population and sort of the gray tsunami that's headed our way. And this is likely to have a compounding effect, not just because of the absolute numbers of aging individuals, but also they often come with comorbidities. So there's concern about how hard this is going to hit the healthcare system. Psychiatric and mental health disorders, that's a huge burden we've heard about here at the conference as well, and the need for novel treatments. And finally, a common theme, and actually something we heard about a lot yesterday, health equity, and the need to address the needs of multiple populations that aren't well served. So with that, I'll turn it back to the panel moderators.
Great, so thank you, Nicole. So as mentioned, rather than going over the 10 unmet clinical needs, which will be displayed and you will see, and the 4 honorable mentions, we tried to distill what are some of the themes that emerge to guide the conversation, and I think as we do that, it'll be touching on the specific unmet clinical needs. The first one to talk about is a systems biology approach to disease. So we are in academic departments divided by organs. You know, we have a cardiology division, a renal division, GI division, and even the way science is funded by government is by institutes that are also organ divided. You know, you will have heart and lung, and you will have digestive diseases and kidney. And of course, biology is much more complicated. As an endocrinologist, I see that a hormone affects many different tissues in the body. So we need to integrate entirety of what's happening inside the organism to really try to see disease. So how do we do that scientifically? Maybe start with Steven Greenberg. How do we take a systems biology approach and harness all the technology that we have, all the omics, to then have a much more concerted approach to understanding a disease process in a human?
Well, thank you. Yes, as you say, medical science has always been interested in systems, whether it's the digestive system, the circulatory system. And I think what we've learned in our approaches to studying systems really changed quite dramatically, or began to change with the creation of the Human Genome Project. So that project completed 25 years ago in 1999. And although it was envisioned as a project to revolutionize human genetic diseases, I think it's had a larger impact on acquired non-genetic diseases because of the technologies that got created as a consequence of mapping the human genome. And those early technologies were microarray technology, then subsequently RNA-seq, and many iterations of that. And we really reached a kind of exponential phase in terms of the ability to learn about human disease by studying tissue through these enabling technologies. And that really created data, lots and lots of data on a scale that we were unable to generate prior to those kinds of technologies. And it's that data which has helped us to try and understand human biology, human disease in tissue in particular, through a systems approach. And I think a lot of that over, maybe 20 to 10 years ago created a lot of novel target discovery, new opportunities to target molecules that we were unaware were pathogenic drivers and how they fit together into other pathways. I think we're close, or maybe have already started to enter sort of another phase, which is system integration or interrogation of human disease by therapeutic approaches that start to dissect these more complex pathways that we have learned about from a systems data approach. Technologies that can target multiple aspects of this. An example I like is in the Th2 space, a very old space of understanding of the cytokines involved in allergic processes, IL-4, IL-5, IL-13. We now have figured out multiple cytokines and their receptors through the systems approach. And there are multiple targeted therapies that neutralize different aspects of these pathways. An example I like is 2 of Pfizer's programs. So Pfizer has 2 tri-specific antibodies, one which targets 3 of these molecules, IL-4, IL-13, and TSLP, and another one which targets also IL-4 and IL-13, but also IL-33. So they're very, very similar drugs, and they're being tested in the same clinical trial in atopic dermatitis, and can really help us understand how these 3 key cytokines involved in allergy differ in very subtle ways, because both target IL-4 and IL-13, and they only differ by TSLP and IL-33 targeting. So I think we're really in a very interesting phase right now over dissecting and interrogating the system through clinical intervention.
So you've raised a couple of things that I may want to follow up on for the other members of the panel. One is the huge amount of data that all the omics generates, and I'd be curious from the private sector, how is that being tackled in companies to be able to make some sense? And then the second piece you bring up is that it looks like inflammation and immunity plays a widespread role in many disease processes in the brain and elsewhere. So maybe comments on both of those. Maybe start with JF or John on the first question.
Yeah, so I mean, from the industry perspective, we have to look at what's feasible. So, you know, some of the topics we have on the list are very laudable and ambitious, but they're probably hard to tackle for particularly private biotech company. So rare disease, of course, is a model, is a prototype of narrowing down the population and looking at effect size, where you can essentially focus moderate resources to demonstrate a significant effect size in the right population. What's interesting is that now we're looking for rare to common, which is how do we address larger things beyond just genetic disease, Mendelian mutation. And for example, in inflammation, as you know, was just mentioned, there is opportunity for small to medium-sized company based on genetics. So for example, in atopic dermatitis, I'm going to follow up on the topic since, you know, Steve brought it up. There is a familial form of atopic dermatitis called Nesserton syndrome. And you know that the upregulation of some proteases in the skin are causing barrier dysfunction. So some companies are going after that pathway, which is distinct from the Th2 pathway, and actually can go beyond just the effects that you can expect from a Th2 inhibitor, such as Dupi and Lebri. And so that would be an example of taking the models that we've used in rare disease based on genetic validation with a familial form and going after the non-familial form, which is a much larger population. But for us, it's very important to have an addressable, doable, contained, endpoints based on effect size. Otherwise, it's very hard to get financing from the capital market. So some of the initiatives that you mentioned on your list are probably more addressable at the policy level, and then we do our part at the industry level, trying to tackle problem in bite size.
Yeah, John.
Yeah, maybe I could build on that. I agree with all that, JF. And from my experience, both in large pharma and now running a small company, which I'll describe in a second, it's that thinking about what could be feasible, but also really parsing out the steps and being very clear about when the project is or is not meeting its potential. And I think that ties nicely into the systems biology discussion, because for me, when we're considering a potential drug or drug target, we have to think very early on, like first day, about how does that connect not just to a single pathway in a cell, but to the entire cellular function, the function of the tissue it's in, the function of the organ, and the entire person, and to start to envision what is the specific effect that one might anticipate intervening in that pathway would have in a patient. And if you can't draw a linear line, at least one line that describes, I think, if I intervene in this target in this way, it would do this important thing for a patient that we could put a number on. You know, a physician would say, wow, that's important. A patient would say, oh my gosh, if you're going to do that for me, it's important. And a payer would say, yeah, I'd pay for that if you can do it. For me, we make decisions that if you can't envision that, we'll find something else to work on. But then it also defines the steps along the way. You need to show data that continuously de-risk towards that pathway, and then you make continued investments. And you know, one of the things that's really broke open, at least in my experience thinking, is the tie into genetics. And I think you're spot on about common disease. I know you were critical in the Broad Institute as well, Jose. But the reason why in pharma we love human genetics, it's a de-risking step. There was an experiment of nature that already told you that mutating that gene and protein in a patient does something important. So that's already sometimes twofold, threefold higher likely of getting to a medicine. So we rely heavily on that. And the last thing I'd say, I agree fully that some of the problems here, you know, aren't ours to solve. They're other people's to solve. But for me, continuing to add to the database of understanding of the complexity of disease is important, because the systems biology could tie it all together, but but there are missing components, and it's really striking what we don't know about biology yet. The company I lead is called Profound Therapeutics. We've got a really interesting platform that's identifying novel proteins that are not encoded by the 20,000 identified in the Human Genome Project. They're completely unanticipated open reading frames. They make proteins that look like they have therapeutic effects or therapeutic targets. And they're adding to the information about how we might help patients. And I'll just conclude, but it's a really exciting time to be doing this.
That's great. I think we should probably move on to the next topic.
Thank you, Jose, and that was a wonderful discussion, everyone. Let's turn to another one of the top three, and that's maybe a global view of infectious diseases with an emphasis on antibiotic resistance. So as we think about infectious diseases more broadly, we've got some issues related to detection. A lot of bacteria we're still culturing. We really need rapid diagnostics to grow in this space for not only bacteria, but for other forms of infection. As it relates to antibiotic resistance, it's estimated by the CDC that there's about 3 million of these infections a year, and that number is going up, unfortunately. And of course, there's many thousands of deaths associated with antibiotic resistance. We have a very small pipeline of novel antibiotics, and there's all kinds of challenges that would be great to get the perspective of the panel on related to scientific challenges, regulatory challenges, and economic challenges, some of which have just been alluded to. Can we prevent antibiotic resistance? Can we outwit the microbes? Let's think about that maybe. Maybe that's a strategy. Also, there's a paucity of host-directed therapies to augment our endogenous microbial host defense mechanisms. Antibiotic resistance has been referred to as a silent pandemic, and it would be maybe remiss if we didn't also mention pandemic preparedness as a really important topic, thinking globally about infectious diseases. The CDC today on its website lists Mpox, polio, Zika, dengue, and Oropouche already as outbreaks, and of course there's concern about avian influenza, other forms of epidemic influenza, not to mention coronaviruses. So there's been lessons learned related to vaccines and those types of strategies. It'd be great to get wisdom from the panel on this. And maybe I'll open this up to anyone on the panel that may want to comment on this rather than targeting an individual. It'd be great to hear your thoughts. Please, JF.
I jump in from the industry standpoint. That's a very painful, topic. Because it's very, very difficult to come up with an economic model where you get rewarded for very good science and novel antibiotics. Part of it has to do with the structure of the healthcare system, the DRGs, where for some diagnostic, the potentially novel innovative antibiotic is lumped into an episode of care and a DRG group. And so that has been a big impediment, and that's probably one of the reasons, and John probably knows more than I do about that, why big pharma and small companies have been very reluctant to go after that, even though scientifically, going back to what Steve was saying about system biology, there are the tools to identify some very interesting target. It's just that the financing and the outcome from a business standpoint is unclear.
Yes, please, Rox.
Well, I'm Rox Anderson. I'm a dermatologist, and I'm privileged to be a dermatologist that defines that as the skin and everything in it. So systems biology sort of is our life. And I just have to inject some positive thinking into this discussion, because I think the paradigm of well-targeted pathways of a new antibiotic is easy for the pathogens to overcome. And so we really need to think more broadly than that. There's new data from the center that I run that you can actually use photons, to reorient the antibiotic sensitivity of multi-drug resistant organisms. There's lots of other strategies afoot that are not small molecule driven, but are sort of biology versus biology, bacteriophage, et cetera. So I think it's a great grand challenge that we better meet, and I think we can.
Thank you.
Yeah, I could just jump in for a second. Not so much on sort of the technology or the science behind it, but just to echo what I heard in the interviews of what lies ahead, that, and not to bring us down into a depressing space, but that it's projected that by 2050, more people will die of antibiotic-resistant infections than any other cause, including cancer. So I think it is really a problem that we've got to put our heads together on and figure out some solutions.
And one element maybe I'll just comment on is, if COVID wasn't a wake-up call, nothing's going to be. And a lot changed, but a lot didn't change in terms of the perceptions and the business model. So I think there's an opening there to push. Part of it I think is also us collectively getting clearer about how we define some of the endpoints and the opportunities. Because I think there are opportunities to define them in ways that might be commercially viable, and if you could sort of pick away at that, we can make a dent, but there's a lot of work to do there.
Thank you very much. Let's maybe move on to the next topic.
That's great. And on your screens now, you're seeing the top 10 ranked by the way they came out in the algorithm with the 4 honorable mentions. So I encourage you to take note. But again, we're distilling themes that are common to these 14 unmet clinical needs. So the next one really has to do, we talked about data already. Data is generated through omics technologies. But there's also another huge source of data in real-world patient outcomes and epidemiological data sets and healthcare systems. So Mass General Brigham, of course, has an enormous biobank that leverages all of the clinical information that we have accumulated on our patients and then ties it to genomic information in a large subset of patients who have consented to have their genome genotyped and even sequenced. And the same thing occurs in the UK Biobank and the All of Us cohort of the United States and a number of other biobanks that are either nationally based or healthcare system based. And so the question is, how do we leverage that huge trove of information to maybe segment specific patient populations, maybe those that are most vulnerable, maybe those that are underrepresented in other places so that we address health equity, and then how do we leverage novel techniques in artificial intelligence, machine learning to distill the data that can be then fed not only to academic investigators, but everybody in industry so that we can move the needle. And I think that we'll open that. Maybe start with, well, all of us here can answer that. Who wants to take a stab?
I'm happy to jump in because, again, as the voice of some of the people that I interviewed. But I think one thing is to really be intentional to make sure that the data that we're collecting fills some of the gaps that we know that exist. And those gaps span a number of important populations. But a little self-interested one is women. I heard from people in my course of my interviews that there's some glaring gaps just on the basic research side. At least well over half of women will give birth at some point in their lives, but that we don't study models in the laboratory typically that have given birth. And similarly, rodents don't have a human equivalent of menopause. And so just being really intentional and making sure that the data that we're collecting covers these understudied populations is really important.
So from our industry representatives here, how does industry try to have access to clinical data, for example, to inform decisions about therapeutic pipelines or target discovery?
Yeah, so it's actually a very interesting topic. All of us have access to the public database. I think most investment firms and companies of a certain size are essentially getting access to those and willing to pay. The issue is the density of the phenotyping, right, which is, we all know is not dense enough in many of those databases, UK Biobank, for example, depending on the phenotype you pursue. So you need to cross-correlate with other databases that are actually private. And that's where the healthcare system is very important because some provider network actually have very good phenotypic coverage. Now the question for them is the other way around, which is how many patients actually accept to be genotyped. But then when you start to cross-correlate some of those databases, then you can actually look at very interesting association, looking at protective allele or risk allele, and then look for where you might have enough effect size that you can actually justify an investment in a company and then going after a clinical program. So I think we're all looking at that, and it's happening in real time. The question is the access. And different healthcare systems have different views in terms of what is the value of their database, even though the database itself as a standalone may not give you all the answer. You need to correlate with other databases. And so it's a bit of a puzzle, a matrix of potential deal and strategic outlook as to how you can collect the data and actually identify those novel targets, whether it's system biology or going from rare to common, where you actually can find a path to a new indication or at least to a new intervention.
Of course, one of the big, sorry. I was going to say one of the big advantages of the clinical data sets is that they're longitudinal. So
Yes.
you have a lot of outcomes, spanning decades sometimes.
Yeah, that's exactly where I was going to go. That's the single piece that's extremely informative and almost uniformly missing from existing, well, it's often missing from existing databases. And yeah, for me, this idea of identifying specific populations and like a personalized medicine approach where you identify a specific mechanism in a particular subset of patients and having the courage to go after that. And I think the pharma companies are now becoming very comfortable with not going for the big market. Actually, your market's bigger if you identify the patients in whom there's a bigger effect size and a cheaper study and you get to the market faster. And then maybe you have a potential to expand. But we need to do a lot more of that and break away from these 19th century definitions of Alzheimer's disease and the like and really molecularly subset the diseases.
And I think there's been a lot of infrastructure to combine the systems biology with the systems clinical data here, particularly in oncology. I think a lot of cancer centers have put a lot of effort into interfacing, collecting detailed systems biology data on all patients at all visits, coordinating that. I think the issues start to become how to share that data, outside the institution, even with inside the institution, how do researchers get access to that data?
So there's a number of barriers that need to be overcome, so that patient confidentiality and their privacy and even their own desire is kept, but at the same time, we leverage not only within the academic world, but beyond the academic world. And I would say there's probably even more of an effort on an ethical perspective for the vulnerable groups, we have seen maybe their data used in ways which they had not consented, and it's not only a decision about an individual when that data is analyzed, but it's about the entire group and their understanding of their own identity. And so making sure that patient groups and advocacy groups are involved in these conversations, I think, is absolutely essential if we're going to make any progress. Should we move on to the next one?
Sure.
Okay.
Great. Thank you. One of the other big cross-cutting themes was improving health and healthcare across key populations. And maybe I'll just highlight two extremes here. First, starting with geriatrics. The most significant demographic challenge globally is now population aging, not population growth. And by 2050, it's estimated that the number of adults in the US greater than the age of 65 will double. So we have a lot of challenges to geriatric health, managing geriatric independence, and thinking about geriatric healthcare, of course. If you're older than 60, almost everybody, it's greater than 95%, have at least one chronic condition. And more than 75% have at least two. So maybe we could think as a panel about how we might approach health span as opposed to lifespan as it relates to the geriatric population. Do we need to redesign care pathways, telehealth, remote monitoring in the home through devices and the like, hospital or rehab at-home options? So I'd love to hear the panel's thoughts on this. But maybe before I open it up to the panel, let's think about childhood and pediatrics. And this might bring us back to the rare disease discussion that JF and John started with. Remembering that a rare disease is defined by less than 200,000 folks in the United States with the condition. There's more than 7,000 identified rare diseases. 70 to 80% of them are genetic, and they're often first detected in childhood.
I'd say sort of social aspects that Rox referred to that probably need intervention, and kind of the reductionist approach of let's find a new target might not be applicable. But that said, there are certain aspects of like diseases of aging that have been difficult for pharma to break into. For example, muscle disease, and it's fairly straightforward to think about. Elderly people almost universally, and particularly accelerated in women, use muscle mass over time as they age. But yet it's been extraordinarily difficult to bring medicines forward that promote muscle health or muscle mass. And it's in part because sort of the endpoints aren't clear, the regulatory strategy is not clear, and it makes it a barrier for investment, even though there are some very interesting targets. So I think there's progress to be made in geriatric medicine and pharmaceuticals to really think more critically about endpoints and regulatory strategies and being a little more flexible in thinking.
Thank you.
An interesting example here is that you probably know that, John, but Novartis had a drug, an antibody called bimagrumab that was tested in cachexia in older people, and they had a signal, but it was not strong enough to move forward. And that was in-licensed by a biotech company because that drug was also actually causing some weight loss, both fat mass and lean mass, which of course in older cachectic patient was not exactly one of the effects you might have been looking for. But it turns out that in obese patient, you can promote some weight loss without muscle loss because it was actually a cachexia drug. And so that's actually moving forward in a different indication, different population. But it underlines the challenge that John was pointing out, is that the signal was actually not quite strong enough to move forward into an elderly population.
I think the other challenge is, for the gray tsunami is recognizing that some of, what we call normal aging and are resigned to may be within our control to change. So John brought up loss of muscle mass, which the industry refers to as sarcopenia. I'm a neuromuscular specialist. I specialize in diseases of muscle. We don't have that. We don't call that a disease. I don't see patients with sarcopenia, but it is a problem. But we call it normal. We don't recognize it as even a disease to draw our attention to. So that's a real problem, I think, to address with the gray tsunami, is sort of recognizing that we can possibly control some of the aspects that we just don't call disease, we just call normal.
That's why I was going at the regulatory endpoints, to recast it as something else, helping the patient in a different way, even though they don't have, a disease.
Mm-hmm. Right.
Excellent, excellent. JF, could we, since you started maybe alluding to the rare disease, and both you and John have mentioned this a little bit in some of your earlier remarks. Could you expand on that?
Yeah, so, rare disease obviously has become, as you all know, in the past actually 2+ decades, a preferred ground, particularly for small to medium-sized organizations for obvious reasons. Population size, well-defined, prospectively genetic marker, large effect size, and of course pricing. But what has been interesting, though, is that has essentially promoted a huge investment in novel modalities, because to intervene in a rare disease where you have loss of function or gain of function, typically it's very hard to do with a small molecule. It's hard to have a small molecule agonist or a small molecule that is going to essentially abrogate the expression of a gene by 100%. And also it's not a very friendly modality for the patient, even though it's orally available, but you have to take the drug every day and so on, and again, you don't have 100% coverage. So genetic therapies, starting with gene therapy, AAV, going back many years, which now has reached some limitation in terms of persistence and tolerability, has been now, I wouldn't say replaced, but has been followed by gene editing, both DNA, mRNA editing, and then, of course, delivery of oligos, where you can essentially, and the challenge, of course, in rare disease is that initially we could only deliver oligos mostly to the liver. And one of the biggest frontiers right now in the industry is being able to deliver oligos to all compartments of the body. So going back to a system approach, going back to expanding the scope of what we do. And I'm very encouraged that there are a lot of progress in terms of delivery of oligos to the CNS, to the muscle. We've seen that recently with exon-skipping drugs and DMD, DM1. And so I think that in this investment that was enabled by a smaller subset of genetic disease is now going to spread out to a larger set of genetic disease in multiple organ systems. And as the safety, tolerability, and understanding and cost of those modalities comes down, I suspect it's going to be extended to not so rare disease. And I think you should expect that gene editing or some form of gene editing over the next decade is going to start being applied to diseases that are not strictly Mendelian disease. So it's going to be very interesting to watch how we're shifting modality based on the enablement of the original investment through rare disease.
Thank you. Any special considerations thinking about pediatric patients that anybody would like to comment on? Rox or others?
Well, I admitted to being a pediatric dermatologist. Kids are, as was mentioned, they are the point of entry for many of these rare diseases. And like right now, my lab, we're working on neurofibromatosis type 1, the #1 disease of humans that is due to a single gene. And the kids are looking normal at birth, but their big problem is by age 20 or 30, they're grossly disfigured. They're hiding. You don't see them out here running around. And that sort of brings me to a point, which is to come up with new innovations for the quality of life, right? So rather than being, the tumors they get don't kill them, they just ruin their life, right? So we've come up with some non-invasive ways to inhibit those tumors from ever growing. There's multiple examples like that you see. We haven't really talked about scarring, which is on the top 10 here, that when a child suffers a burn injury, the kid tries to grow, but the scar is trying to contract. It's a primitive wound healing mechanism. And you just see this horrible morbidity associated with that. I think we need a National Institute of Fibrosis, just like we need the cancer institutes in heart, blood, and lung. And fortunately on that, there's really some new hope. I mean, I think the YAP-TAZ pathway, as it's being figured out, and mechanotransduction, the gross abnormalities of scars can actually be turned back in. We see this in children, by the way, with burn injury, that you can tickle those scars to normalize themselves. So the question is, could you do that in other organs? So we're almost out of time, but I'm pitching for derm here. Derm's a great playground, right, for the rest of the body.
Thank you.
All right, so as predicted, we are out of time. There's a lot to digest. Of course, you have all the topics on your screen. Maybe before we close, we can have one final word from each person, 10, 15 seconds from each. You can say whatever you want, but if you're looking for something to say, I would say if you had unlimited resources, unlimited funds, you were a government or you were a company that was just newly endowed, what would be the one area that you want to go after on the things that we have on the screen? But you can say something else if you don't want to answer that question. We'll start with Dr. Greenberg.
Well, besides for world peace, fibrosis is, I think, an area that cuts across many different diseases, is quite refractory. It's a end point of autoimmune disease, inflammation. We really need better medicines in fibrotic diseases.
Thank you. John.
Well, look, I do have a company, so I'll take the money you're offering. But look, I'll just kind of come back to working together to, while we're solving some of the social problems and things that are addressable by governments and healthcare systems, to also just keep pushing on redefining diseases by molecular mechanisms and having the courage to subset them. And I think we'll make substantial progress. I'm very enthusiastic about what can be done.
Wonderful. Jeff.
I'll do very quickly a policy one, which is reforming the healthcare system in a way that more funding makes more sense for more opportunity. So there's tremendous amount of money being wasted right now. And two, on the science side, a Manhattan project of data sharing across all type of data, public, private, genetic, phenotypic, longitudinal, endophenotype, anything, and available to all players, so that we all have access and we can compete based on that enormous pool of data.
Wonderful. Nicole, you're the voice of Harvard Science, so go ahead.
Well, I was going to say, I don't have a company, but I'll take the money anyway. And it's hard for me to choose one of these, as I feel like they're like all my children. So what I will say is a little bit more general, but I think continuing to have forums like this and engage in projects like the Unmet Needs to really have people come together, think about new ways to collaborate and solve some of these super big problems that aren't going to go away anytime soon.
And Rox.
Well, if you give me enough money. So I live at Mass General Hospital and I run a research lab, and I'm not dinging anyone here, but there's two hospitals. There's the research people and there's the clinical people, and I think we have a huge opportunity across the country and the world to bring those two together. You have a bunch of people using today's technology facing problems that they can't solve. That's the practice of medicine. And you have a bunch of people that are incredibly smart but don't even speak the language that the clinicians understand.
Yes.
So give me the money.
Well, thank you. Those are transformative suggestions. I'd like to thank my co-chair, Bruce Levy, and then our panelists for a wonderful discussion. Thank you for joining us.
Thank you.